Hello, my wonderful biology humans. Welcome to semester 2. Yay, you did it. Woohoo! Halfway through the year. Okay, so finishing up topic 2 is going to take us a while. All this biochemistry. And then after topic 2, 3, and 4 are relatively small. And we have definitely a lot more labs, bigger activities this semester. All right, so 2.4 proteins. We've already basic, like, functions of proteins, basic buildings of proteins. But just to go back, and I want you guys to have some things in your notes. So first of all, this is big picture right now, so you don't need to write any of this down right here, right? But this is good to look at. Like, this is kind of like the big picture of what we're going to be covering. Don't need to write that down, but things like this you do need in your notes, right? And I have this on the next slide, but we are going to be looking at like the molecular level of proteins, just like carbohydrates and lipids. So how they're built, how they're put together, and then the bigger picture of how that plays out in life, right? So what are they used for? How are they broken down? Things like that, right? So how do we go from these tiny little amino acids being linked together to these proteins that do everything in our bodies? Okay. All right. So let's just go ahead and start out with some basics about amino acids. We're going to start small and build up to the big picture. Okay. All right. So amino acids, I want you to go back a little bit to like freshman bio when you learned about amino acids, and I'll show you in just a second. But basically, your acids, your mean amino acids, are linked together via condensation, right? So this is one of the things that you will have to draw. I'll show you that in the next slide there. But it is something that you will have to know how to link the two together and draw a condensation reaction, and we will do some practice about that in class. So remember that condensation is when a water molecule is it comes out, right? So is removed when two things come together, right? So condensation is going to be putting two things together to build up bigger molecules. Hydrolysis is when we use it to break it apart. All right. So going back to our organelles, when we looked at like what do ribosomes do, right? Ribosomes make proteins. Well, now we're learning the nitty gritty about how ribosomes make protein, right? So condensation, that reaction occurs on organelle that we've already talked about, which is the ribosomes, which are either free floating or in the attached to the endoplasmic reticulum, right? This whole process is called translation. We will learn a lot more about that later. So just for now, know that translation is when we translate information from our DNA into the language of proteins. So I have this like secret code in my DNA, right? The different bases put together in different orders. And the translation part is when the actual amino acids are linked together to make a protein, right? So that process is called translation. We're going to do a ton of work on that later, but just kind of know that word for now. And the certain type of link that occurs when they're put together as amino acids is polypeptide. So we've already had exposure to this word. Might be a little bit of a review, but we will be going back to like our root words of like, what does poly mean? We've already seen poly, right? And polysaccharide. And then what is a peptide bond? How do we draw it? Where do they occur? And what is like the end results? Okay. So a polypeptide is when I get my tiny little amino acids and they are linked together. That is the type of bond that occurs, right? A peptide bond. And when I have many, right, it's the poly. And then some of them only have, so some of the proteins have only one polypeptide. So put together over and over, and some of them have more than one. We're going to be looking at those as well. All right. So for drawing purposes, here is that reaction. Okay. So some things that could be a review or might be like the first time we're hearing it. An amino acid, a peptide bond is formed. And so make sure you have these things written down. When a carboxyl group right here, right? So I've got my carbon, my hydrogen, and my oxygens and my hydrogen. This is a carboxyl group is going to make a bond with an amine, right? So this is an amine, this is a carboxyl group, and those two come together to form a bond right here. So you can see right here, so if I kind of look down here, right? So I've got, this is the old molecule here, right? So there's that. And then here is the other molecule here, right? So that is this one here. And so when you see what's left over, I still have that oxygen, right? the double bond oxygen, but the bond that the carbon used to have with this oxygen and hydrogen is broken, and it is going here, right? So it's attached to the nitrogen now. So look at the products of that, right? So this right here goes away, and one of the hydrogens go away, right? And those two come over here to make water, right? So if you can kind of remember when you have these two things drawn, right? So here is my carboxyl group on one end, my amine on the other, and those two come together to make a new, right, a new structure here. So this is called a peptide bond when the carboxyls and the amines come together. Okay, so we'll do some practice with that in your book. You will need to know how to like draw this simple. Oh, I just said simple. This reaction and this process. And we'll do more practice with like if I'm given an amino acid, right? I've got these two amino acids. Remember that the basic structure are these amines and the carboxyl. The R is what makes them different, right? R is that differentiates the amino acids from each other, but that they all have these basic structures, and this is how they come together is through a condensation reaction between the amine and the carboxyl group. Okay, so I go from having my single, right, to my dipeptide, and then we're going to look at my polypeptide too. Okay, all right, moving on. So things to note about this reaction. I basically just said those things, but now you can make sure you have them spelled right, and you have those actually in your notes, right? And so the bond is formed. We've already looked at that between my amine group, right, and the carboxyl of another. I get that H2O that is removed during condensation, and that new bond that's formed is called a peptide bond. And then here are kind of where I'm going to bring in my prior knowledge. So di meaning two, right? Poly meaning more than that, so more than two. And then we do have this little kind of special group of peptides that are called oligopeptides, and they're sometimes used for chains for fewer than 20 amino acids. So these are different type of peptides. Those 20 amino acids that our body builds, or I'm sorry, just cells build all proteins from, we're going to look at in a second. So but we'll look at history in just a second, right? So just know those two for now, right? So I'm sorry, 3 dipeptide, poly, and oligo. So make sure you write that down. There we go, and have that language in your notes. Okay, just kind of this is in your book as a little like side note, looking at the differences in our like how big a chain of amino acids could be. I think I opened your book earlier. I did. Yes. Okay. So here's like looking at how different. So here's where we're going to learn how to. I'm just like looking to put together the different type of amino acids. And there is this little box on insulin. Oh goodness, I can't even find it right now. Anyways, it talks about like the difference. Oh, there it is. Scurvy and then insulin. About what like the sizes of the different proteins that are made and the different things that they do. Insulin is actually a super cool molecule that we get produced. You've probably heard of it with diabetes, that is produced in all organs. We can actually use it because it is a protein that is the same in all living things. It can be like produced in one organism and then given to another, right? So one of those other exceptions on the other end, so insulin's a very small one. Titin, which is, I'm sorry, I think it's not Titan. I've heard it pronounced Tintin. I'm sorry. You can look it up on the dictionary. It's in your muscles, right? So it's a really long amino acid chain in your muscles. So this one, right, is like 121 amino acid and then 130 put together and make one. This is over 34,000. So just know that there's a wide range of proteins and how big they are based on the amino acids putting together. Okay. All right. So on page 88, this will be something that you're doing in class, right? So you're doing this in class. You're taking these notes on Monday because I am not here, right? So this will be your activity. You have a separate piece of paper in here, as your activity. So make sure you draw these things. So on page 88, there's that purple box, and it's going to ask you to form different peptide bonds between these. Go back in your notes and look at how the carboxyl and the amines come together, and make sure that you're able to draw those on your activity box. And we can go over those on Tuesday if you have any questions on that. Okay. All right. So looking at, there's that circle I need to get rid of. The 20 different amino acids. Okay, let's go back. Don't look at that chart yet. Do you recall this chart right here? My codon chart. Remember this a little bit? A little bit, kind of. So you saw codon charts in different ways. You might have seen it like this as a chart in your book. You might have seen it like this. But if you recall, and if not, it's okay, we'll be going over it, that my 4 different base pairs, A, T, G, and C, then go to A, U, G, and C when they're going out to make the ribosomes. And based on the 3 codons that come, the 3 bases that come together make a codon, these are my 20 amino acids, right? So all the way around here, these are the 20 different types of amino acids that are going to be made with the 4 different base pairs. So so I've got 20 different possibilities from here, and then you can imagine that just I'll show you like millions of different possibilities that can come together from linking these together, right? So these are my amino acids. So based on what the DNA base is, that is going to produce my different type of amino acid. Okay, so going back here now, right? So I've got those 20 different amino acids, and those are synthesized on ribosomes made into polypeptide proteins. Okay, do not write this down. Do not write this down. Do not write this down. All right. This is shown a little bit differently in your book, but you should know that the amino acids are broken into different types of groups, right? So you do not need to memorize these, but you do need to know that there's nonpolar, right? There's aromatic, there's positively charged. Oh, these are all based, on their R groups, right? The things that come off. So look up here, just again, real quick as we're looking, right? These right here, same structures, right? I've got my amines, I've got my carboxyls. So this is all the same. This is like the base at which it's built. And then this on the end, my R groups, right? This is the group that comes off. These are what makes them different, right? So nonpolar, polar, negatively charged, positively charged, and aromatic, meaning that there's that carbon ring there. So now that we've seen more examples of like, identify this molecule, right? So now you can see, oh, anytime I see this amine and the carboxyl have come together, this base here, this is going to be an amino acid, is going to be a protein. Okay. All right. This is shown a little bit differently in your book, so pay attention to that box as well. And I'm pretty sure, yeah, it's right here. So it's different colors, right? So here's the boxes without the words in there, right? So without the amino acids, you can see that those are broken up into those different types of groups. Okay. All right. Let's see. And here we go. Okay. So another one of those little boxes, scurvy. Type of ascorbic acid is needed to convert, these are all amino acids, prolines into this hydroproline. And so if I have a deficiency in that amino acid, it's not able to produce collagen correctly, right? And so collagen, right? No some collagen, what it does with your skin, the structures, different like supports. I want you guys to look up scurvy, right? And then see what you think would happen, like how scurvy is caused, right? What are the symptoms associated with it? This is also on your worksheet as well. Okay, so look at like who suffered from it a lot, why they suffered from it, so what they weren't getting in their diet, and how that could be possible. So make sure that's on your worksheet as well. Okay, a little bit about the history of amino acids. So going back to like, where's the evidence that we have on where these things came from. Oops, sorry, I pressed the wrong button there. Most organisms use the same 20 amino acids, right? So one of those things that we've already looked at with whether or not something's related is, remember the differences in amino acid base. Like there's that chart, like a fruit fly has this, right? And then a human has this, and a monkey has this. So looking at the difference, but that's part of that evidence that we used in Unit 5 or Topic 5 to look at like common ancestors, right? So here's the different hypotheses you need to know about the amino acid origin. You need to know that the 20 amino acids were the ones that were made on Earth before life, right? So we found these amino acids around even before they came together to make small molecules. And they were the only ones that were used when life began, so they put together things. These 20 amino acids are ideal for making a wide range because they're able to make those peptide bonds pretty easily, and then they're able to put together in different ways, different orders, different ways, and build different ways, and that all life evolved from that single species, which used those 20 amino acids. Right? And so we've looked at those as evidence for common ancestors, for looking at what evolved first, but that's giving you a little bit more background on the amino acids. Okay. All right. Let's go. You know what? Here, we're going to stop there. What I'd like you guys to do for the lecture for now. So now we're going to go into how those amino acids become the proteins, right? So I'll show you this one for now. But so if you can start looking at like, okay, I've got these 20 different proteins. Again, here's my amine and my carboxyl. Got these 20 different amino acids, like different orders, different things. And we're going to talk about how they're going to start coming together to make different structures of proteins. So tertiary, primary, quaternary, different structures of proteins. But we'll stop there today for amino acids. So go ahead and keep working on that worksheet. You have your notes for today. You've got those drawings. And then tomorrow we will talk it back more into like how they're built into proteins and all the different functions. We have more functions to memorize. All right, you guys have a fabulous Monday, and I will see you tomorrow.